Okay, hello everyone. Hello Ben. It is a great pleasure to have you here at my channel. We have agreed to chat on your studies of the Talin protein and the MeshCODE. And I have been having a conversation with Speed or Mike at his YouTube channel. So afterwards, I contacted you to have also a conversation. So maybe you could quickly introduce yourself and say what your investigation is in biochemistry and science, and then we can go from there on and have a conversation.
Sure. Okay. Well, thank you for the invitation. Yeah. So my name's Ben Goult, Dr. Ben Goult. I'm a reader, which is an equivalent of an associate professor at the University of Kent here in Canterbury in the UK. So just about an hour outside of London. And my research for many years now has been trying to understand how the cells in our body, in fact, all cells, can sense and feel physical environment and how they can convert physical signals into biological signals. So we're mainly a biochemistry lab trying to understand the building blocks and the proteins which mediate this, with a main focus on the protein talin.
Okay, I have actually dived a little bit into that protein. And what I understood is that in order for it to act and to work, it needs to be in a work in a joint venture with many other proteins, like one to the other. Is that about correct?
So that was a bit crackly, but I think, yeah, if you turn about maybe a little bit about what these proteins are involved in. So talin's an essential protein for the development of multicellular life, because all the cells in our body are attached to the correct places via engagement with each other, with the neighboring cells, but also to this surrounding material called the extracellular matrix. And all of the cells attach onto that, and they do that via a family of receptors called integrin proteins. And the integrins are like the hands of a cell which grab onto the matrix. But just inside the cell, you have this complex protein called talin, which makes a direct link to the cell's force generation machinery and to the motor proteins which are inside the cell, which can be switched on to generate forces. And into that simple core of an integrin and a talin molecule, and then the cytoskeleton, it's called, this machinery, up to 250 different proteins can also be recruited to that site depending on the physical environment, what that cell's doing. So there's incredible complexity in these adhesions, they're called, these integrin adhesion complexes, and talin's the core component of those.
Right. So basically, they have to work in an integrated fashion together in order for there to be functionality. Is that correct?
Yes, I'd say so. So the integrins, as they're shown here, they can switch between an inactive state and they can be switched on. When they're switched on, like as in the drawing you've got here, they can grab onto that cargo net outside the cell. And talin is recruited to these sites and switched on when it gets there, and that switches on the integrins, and then you get an adhesion. So if talin lets go, the cell will let go of its attachment. So you can control whether the cell is attaching or letting go via the signaling pathways inside of the cell. And this is quite a unique thing. It's called inside-out signaling, where the signaling inside the cell can send the signals outwards to let go and grab on. And this is very important during things like cell migration, where the cell's crawling along, climbing along this net. It controls it from inside, from its internal machinery.
So let me show this image here from another paper.
That's actually a paper from Tony Kanchanawong, and it's actually a slide out of one of my seminars. I recognize the header, but it was to try and summarize this basic idea that these attachments which the cell makes are really complicated. So at the bottom of that image, they've got this blue layer and this sort of light blue layer, that's the matrix. That's what the cell's grabbing onto. And those red proteins, they're the hands of the cell, where they're the integrin receptors, physically grabbing onto that extracellular environment. And then got the membrane, which is sort of between the red and the yellow line. That's the outside, that's the plasma membrane, the edge of the cell, the lipid membrane. And then in blue, just skipping out, you've got the cytoskeleton in blue. This is the actin, this is also microtubules, but it's also got the motor proteins, the myosins, and they can pull on that cytoskeleton, those filaments, and pull and generate forces. And what we work on is the yellow and green bit in this sweetie diagram. And the yellow is the talin. It's essential to make this linkage, but it also serves as a computational layer, because what I've not mentioned yet is that this talin protein, not only does it attach these two together, the integrins and the cytoskeleton, but it also contains a complicated network of series of binary switches. I don't know if you've got a nice figure of what talin looks like.
Yes, let me scroll down here. If you want to share, if you have anything to share, feel free, just tell me and I put it on the screen, okay?
Okay, sure. So this one's fine. These are most of my stuff anyhow. So this is what the talin molecule looks like. At the end, you've got this firm domain. That's what grabs onto the membrane and the integrin tails. And at the very C-terminus, where it says R13DD, that's where it attaches to the cytoskeleton in the simplest version of this. And the bit between R1, R2, R3, these are these mechanical switches. And it was when we discovered that these were mechanical switches that's what transformed our direction of our research. And that was because we realized, so we discovered this via biochemistry, trying to understand what this molecule did. And this protein called REAM in panel A, once we found that sticks onto the side of these switches. So the switches can go from a folded state on the left to this open state. So they can open and close depending on if you pull on the molecule. So you can have it in a closed state bound to this REAM protein, pull on it.
But is there then really that distance from one binding point to the other here? Because here the configuration is closed, right? I don't understand when it opens up. Is there this distance here, or how does that look like?
Well, if you imagine it, there'd be sort of, if you imagine that it was pulling that way, then the bundle would be laying down, and then it would get pulled into a long string, so it would get longer.
Okay, so it gets longer. Okay.
Yes. So I don't know if you've got my MeshCODE paper to hand, but I tried to draw that more schematically in figure 2 of that, where I show what a talin.
The talin as mechanosensitive signaling hub. That's the paper.
Yeah. Have you got the MeshCODE paper to hand?
No,
I think
don't.
It shows it quite nicely.
Let me check if I can pull it on the screen here.
I think it shows, because I think what you're getting at is, what's the distance here? And they go from these compact closed states to this quite extended long state. And that difference, you can switch between those two by pulling on it or letting it go and allowing it to refold. If you've got it to hand, that's great. If not, I can share it. It came out in earlier this year.
Yes, I have it on hand, but I think it would be more interesting if you share it, because then you can go with the cursor over it. And
how do I do that? Share. Okay. Oh no, not share screen. I don't want to do that. Share. Sorry about this. Just give me one sec.
Mm-hmm.
Can I just share the file, or do I have to share my whole screen?
Yes, I think I will have to put it on the screen. You share it, and I'll allow it to be shared.
Oh, great. Oh, screen sharing is easy for 2 monitors, but I haven't got 2 monitors. Okay. Right, just one second. Share. Oh, yeah, this is going to be fine. Oh, man. Sorry, no technical issues. Maybe
No problem. Do you want me to share it?
Well, the funny thing is, it's not in the list of ones which I've got. I've got it open, but it's not in the list I can choose. Maybe. No, I think, yeah, if you share it, then I'll.
Okay, I share it. I just need to know which of the figures you want me to show.
Yes.
Afterwards.
So number 2, figure 2.
Figure 2. Just a minute here. Figure 1, figure 2. Just a minute.
I seem to have crashed this. I can't close that window, so sorry. Technical. Oh, it's here. It's come up. I found it now. Okay, you've got it. Yeah. So this figure 2. Maybe I could share it then. Is that sharing what I'm showing you? Yes. Okay. So this is the MeshCODE paper which I published earlier. So this is like a culmination of all of our research into an idea for how all of this might work. So we've been working on this mechanical coding for 15 years, but it all sort of came together into this theory of a new way of visualizing how information storage might occur in living organisms. But the figure I was hoping to show, and if you can still remember what we were talking about, then it was this one here to try and show how this is all working. So here's the same representation of these 13 switches, but in this panel C, I tried to simplify it just to show 3 switches in a row and to show what would happen if you were to switch the motors on one time. That you would go from this all closed, 3 closed domains to 2 closed and 1 open domain. And the reason for showing that is that this is the open state where it gets pulled long. And if we were to pull on this 010 state again, then it would go to an 011 state where the second one of these other domains would open up. But the reason why this is important is because what you were showing on that other slide, where the proteins which interact with this single domain, in this case this R11, which goes from this closed state to this open state, it binds to different things, different signaling molecules, if it's in that top shape or if it's in this open shape. So when
now let me quick stop you here. Where do the other molecules or proteins bind to sense if this R1 is in open or closed state, and then can actually make up, interpret the information?
Yes, so that's a good question. So the readout of how this, so this is the same thing, but just drawn as a cartoon, because it's easier to visualize. But this is showing exactly the same as what I showed before, but here with 4 switches, 1, 2, 3, and 4. In the folded state, you've got all of these protein molecules which recognize that the binding sites on the folded domain. So the readout for this state is to recruit a green, an orange, and a yellow protein to that site. If we pull on it,
so they bind actually to these sites. Is that correct?
Yes, that's a good. Yeah, so they bind to these sites, but these ligands do. But in this next figure, the blue one only binds to that open state. So when it's in the closed state, this number 2 only binds to the orange domain. But when it opens up, it now switches. It goes from the orange domain to the orange protein to the blue protein. So the signaling at that site, because the key thing to mention here is that these switches, they're like conformations of a protein, but the readout and the actual what happens is that you recruit these different signals to that site, and that's what changes how the cell behaves, because it changes the signaling complexes.
So it's basically the signaling, the information that in one state you have the orange protein, and that signals one state of affairs. And when it opens up and the blue protein binds to the two section, then that means another signaling information. Is that correct?
Yes. And there's two ways that this can happen. One is you bring that blue signal to the bottom of the cell where it can control the behavior. But also we've got examples of things like transcription factors and splicing factors, which, like say this orange guy, so once this domain unfolds, that orange guy leaves the bottom of the cell and can diffuse into the nucleus or go elsewhere. So you can move things around the whole cell as a function of the pattern, whether these switches are open or closed.
So let me understand that. Would that mean that as soon as there is the open state, the orange protein detaches, goes away from Talin, and then it can actually go to another location in the cell, maybe in the nucleus or wherever it is, and then signaling, hey, the state of Talin is open now, do this or that. Would that be about correct?
So, yeah, this is a process called mechanotransduction, how all of the cells, this is one way that they can sense those forces and sense mechanical conditions and convert it into instructions for the cell. So yes, that's one scenario.
So how would that orange protein know where it has to go inside the cell and deliver that information in order to start another biochemical process?
Well, I wouldn't say it knows where it's going to go, but if you think of it the other way around, when there's a binding site at the adhesion for that orange guy, then if he diffuses there or he gets enriched there because he's got specific binding sites there, if they're no longer there, then that protein will float off, and it might either get there just passively, it might just get enriched at the adhesions, or it might hitch a ride on a motor, a microtubule, or there's different ways it can move about the cell. But it could just be that all of these proteins just get relocated depending on where there's available binding sites or interacting partners.
Okay. So what I understand now is that these binding proteins, the signal, it can be delivered at a far distant part inside of the cell. Is that correct?
Yes. And also the other thing to mention about this is that these adhesion complexes, these talins, connect to the cytoskeleton and to the microtubules, and they're really long filaments, so they connect to the nucleus and they connect to different organelles in different places. So the whole thing sort of connects up with these linkages.
Right. So another question. You said that these switches, they can be in a closed and open conformational state, and when they open up, then the Talin protein, it lengthens its size, right? It becomes longer. And since it is attached to the microtubules or the actins, is that the size of the protein also bearing information to the cytoskeleton? And then does the cytoskeleton do something based on that size of the Talin protein?
I'm sure it probably does, but in terms of the thinking, there's quite a lot of, and we've made talins of different lengths and stuff. But there seems to be talin's not the only thing which is forcing these linkages to occur. It's part of a huge system-wide connections, like all of this cytoskeleton in this figure here, all of this red stuff. This is all the actin connections, which is the cytoskeleton, the microtubules. So they all recruit there.
So would you say that besides talin, are there other signaling molecules and proteins also involved making up that mesh of information exchange?
Yes, I think there is. Yeah, definitely. I think and these adhesions are really, really complex and sophisticated because they're sensing the physical environment. And one thing I didn't mention is if you make the extracellular environment more stiff, then these switch patterns are different, and the cytoskeleton linkages are different. And so you can build lots of different things. So the cells both sensing what it's attached to, but also building motor proteins attached to it. So it's a complex system.
Yeah, I have collected 45 different epigenetic codes, Ben. I working joint venture together to create all the complexity that we see in biology, like cell differentiation, cell size, all the exchange of information from the environment to then give the right response, and so forth.
Yes.
Now, one key issue that I would like to understand how you see it is the fact that Talin supposedly had the origin with protozoas or with unicellular organisms. And if we go from unicellular to multicellularity, then I think two key features and innovations had to occur, which is cell-cell signaling and cells attached to other cells or cell adhesion. And
yes.
during
and cell matrix signaling as well.
Yes, and during the short investigation which I made of Talin, it seems to me that both signaling and attaching adhesion is something which is an integrated process. That means the correct adhesion from one cell to the other also depends on information, right? So when I studied about the transition from unicellular to multicellularity about 6 years ago, this was a key aspect which got my attention, is that you need two key innovations simultaneously, which is cell-cell communication and adhesion. So the question is, of course, how could that be a product of just evolutionary change, since there are various things which have to come to start operating together at the same time. And evolution is step by step, slowly, one point after the other. But if you require multiple new functions all at once, how do you get that by a mindless, unguided evolutionary process?
Well, prior to multicellular, there are single cell cells which are starting to generate these different versions. Like looking through like a lot of the sequencing of these lower organisms, you start to see these modules and these machinery starting to appear. And at some of the early things like slime molds and stuff, you can even start to see them transitioning between single cell and then going into multicellular for small parts and then back. So there were like the precursors to this. But I accept that there's a lot of missing, and in terms of our understanding of how this all came simultaneously, there's gaps. But it must. But the how I kind of think about it is that what we're basically presenting here is that there's like the model I'm building to is that you've got a lot of relative chaos, what appears like chaos inside a cell, and lots of things moving around. But if you've got switches, then you can have lots of stuff going on. But at the end of the day, a switch is either in a 0 state or a 1 state. If the whole cell is covered in switches, then the whole cell will synchronize this information. And then if there's an advantage to two cells talking to each other and like them synchronizing that information, then you could start to imagine a link to a double cell. Like, I'm not saying I think this is right, and I'm not saying that this doesn't have holes in it, but just trying to think in terms of that it could happen. Like there could be a real advantage to be able to share what's essentially information.
Right. Now,
it's a really strong selective.
Well, a relevant question, of course, would be in that situation, in order for you to have communication and signaling, you need molecules that understand each other. So let's suppose that you have a cell which doesn't have still that signaling code, and the other has actually that signaling code. How could it transmit the information from one to the other if both do not already right from the get-go understand each other?
Yeah, but we're not talking about, like, even if you're thinking of evolution, you're not talking about one cell not having it and another cell having it. If you've got a cell which gets the machinery and it divides and they happen to come into contact and there's an advantage to them, like that maybe they sink down lower or they move, or there's something where them forming linkages to each other is beneficial, then that would immediately synchronize the two cells to be working in parallel. I'm not saying, again, I've not got a strong, there's big gaps in any of this, because whichever way you think, however you try and explain this.
Right. Yeah. Now, another very interesting thing which I just saw is that Talin has to bind to integrins.
Yes.
And it says that the tail, the C-terminal, the binding site, if it is conserved, that means if you change the amino acid sequence, then it is not capable anymore to bind together, right?
Yes, that's, well, so, it's considered that it doesn't change over in all the animals it's in. But I would also say with the C-terminus, if you're going down, is that module for connecting to the actin filaments in red in here, that C-terminal module is conserved it's in yeast, and it's in well, it's in both fission yeast and brewer's yeast, and like basically it's quite a common. It appeared in single cell and then multicell, and it's the bit. I would also say that Talin predates integrins, so there was like this mechanical computation before in species which haven't yet got integrins. So in my opinion, I think that the integrins provide a way to add vastly more information into these systems, because then you can start to imagine information being inside the matrix itself, how stiff it is, where the attachment points are. So you can actually have orders of magnitude way beyond what we can imagine written into these molecular machines and these molecular patterns. And that's where I hesitate slightly on thinking that this is either creation or this is evolution, because we've got no way of understanding that information. It's way above what we currently think, but it could occur just by, once you've got a cell, it could occur by it being exponential in terms of how much versatility or adaptability you enable by that improvement.
Yeah, I am also quite surprised of the size of Talin. It has about 2,400 amino acids, right? So it is, I would say, a rather big protein, 2,541 amino acids, and also the binding site, the firm domain. I'm not sure now. I didn't find the information how many amino acids it has, but do you know that? Do you have that information of the size of the firm tail?
The firm domain's 400 amino acids, and there's quite a nice adaptation there, like how it came about, where in slime molds, which can make adhesions but not very well, they're quite between slime mold and sponge in terms of because even however it got to where it is, evolutionary genetics is really nice for seeing how things improved over time, how those improvements came about. But in terms of the transition from slime mold to sponges, the Talin, the firm domain, looks exactly the same, apart from the regulatory elements appeared after the. So a as you go into the sponges, so there's definitely, you can see an advancement there. So I don't know how you get to the full length protein, but the addition of that loop, it's a loop, enabled this to be dynamic and regulated. So that I can easily imagine. I don't know how you get to a Talin, but I can easily imagine that is a single exon which has got introduced into the gene of Talin.
Okay, so you said that Talin is extant in slime molds, right? And then you have there already a fully developed a firm tail. So I mean, what function does Talin have in slime molds?
Well, slime molds make adhesions, they migratory, because the other thing that these complexes do is they're involved in moving around. They can, because you're basically grabbing onto a surface and then you're pulling yourself forward with the muscle, like how we would climb up a ladder. So the cell is doing exactly the same thing, just making these complexes, pulling against them and moving itself forwards, and also holding itself in place. So
yeah, the difficulty which I actually have is to imagine that there you have eventually a unicellular organism which doesn't have the ability to attach to different surfaces, and then somehow it had to develop Talin and other adhesion molecules. And if we see just Talin with 2,400 amino acids, now let's suppose that it started much smaller. Let's suppose only with 300 amino acids. That is still quite a big number of amino acids which would have to line up in the right sequence in order to get functionality. And if you go lower and lower and lower, there will be a threshold where you will not have any function, right? So the problem would be, as I see it, is to go from 0 to the smallest protein, which then would bear actually any advantage to the organism.
yeah, so even more mysterious is that, like, with the exception of these regulatory elements I mentioned a minute ago, Talin's always been the same length since it appeared. It's never changed. The switches are in the same order. It's been incredibly well conserved. So when it appeared, how it appeared, yeah, I don't know. It's not, because most proteins you can sort of identify how they might have come about, like, and they're like improvements or alterations in something else, and you can trace it back. But to my knowledge, like, most of the Talin's been like the same since, as far as our records and our, as far as we can go back. So I agree. Where does it come from, and why does it? The fact that, like, all of the other big adapter proteins, well, not all, but most of the other big adapter proteins, if they're only connecting two ends together, then it doesn't really matter if they get shorter or longer over time. But but Talin's always stayed the same length, because even the loops I told you about, they don't affect the overall length because they're loops. So yeah, mysterious thing.
That's very interesting information, Ben, and I think it really should put a question mark to those which constantly say, well, evolution is a proven fact, it is a theory, things and that. But when I talk to professionals and scientists like you, and then go actually to the details and to what actually the evidence shows us, then the situation and the responses are quite different. Then actually it is exactly as you said, like now, I mean, we have no evolutionary trajectory and evidence that it went from small to what it is now. We see it since the right beginning, the same size. And that, as you said, that puts a question mark in regards of its origin.
Apart from, like, maybe, like, there's nothing's not like there's nothing before in terms of which is ever like, because the thing with the switches are they've never changed in what we can see, but they are the same domain over and over again multiple times. So it's almost like they appeared in very quick succession, bang, 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 like, and then this was like an optimum length. But that's not as satisfactory in terms of how it would actually work. But it also the repetition and the possibility of basically duplication, and then it just being like, who knows? I mean, we've got a big black spot here, and you can fill it in with.
I understand that you, as a scientist, are fully focused actually to unravel the operation, how it works, and I think this is a fantastic job that you are doing by unraveling this. And of course, I, as a theist, I try to understand how it could have occurred and emerged. So there is a philosophical aspect in regards of that, where the entire dispute between evolution and creation lies in, where those which propose evolution say, well, evolution explains these phenomena, what we observe in nature, and I, as a creationist, I say, well, there are gaps. Now, I could say, well, because there are gaps, therefore I insert God in it. But the question is, are these gaps not becoming bigger and bigger and putting a bigger and bigger question mark in regards of evolution, of if evolution actually is an adequate explanation?
Well, yeah, I don't know. So I can imagine, I'm not saying I can, like, from a cell which has got this machinery and got these workings, I can imagine how you could go from a single cell into a couple of cells, into a cluster of cells, start to specialize. Because if you can capture huge amounts of information in the shapes of the molecules, and that can put a really strong selective pressure on. So I'm not saying it's definitely my number, like I can prove this, but I could imagine from a single cell going to a more complex thing, and then growing. So that bit is, you have to really stretch your imagination. The harder bit, in my opinion, and this is purely my opinion, I'm not saying it from any real basis, is how you get to a single cell in the first place. Because it's like, especially a single animal cell, because it's got all of this machinery, all of these elements, all of these motors and all of these switches all working together. So that's the complication. But from a cell, I think I can imagine a way to optimize and tune this but with a much more selective pressure. But I don't know whether there's strong. And if you look at, in fact, if you look at the evolution of the genes through the evolutionary time, you can see the changes, and you can arrange all the animals in line based on the changes to the DNA. So that would be some evidence that this, from that initial single cell to here, the hard bit is from the single cell beyond, like earlier.
Right. You can see here, I put on the screen number 30. That would be Talin signaling. I did insert that after I listened.
30.
Yeah, I did put it. Actually, I inserted it after I listened to your conversation with Speed at his channel. I didn't have it before, but I am listing here 36 different signaling molecules and mechanisms which direct at least 15 force-generating molecules through at least 39 different mechanisms. So this is a huge complexity inside of each single cell, which then is involved in cell differentiation. I mean, I put it here. Morphogenesis of each cell. This list here is in regards of each single cell. So when I hear people talking about evolution and not going into the details, then I think it is a vain conversation, like looking at fossils and things like that. We need to look into biochemistry and in things like what you are studying, which is Talin, and single molecules, which are really the actors which define organismal complexity architecture, which starts with basically the architecture of each single cell. And Talin is one of these actors, but I think that we can probably agree on that biochemistry and biology is far, far more complex, and we are not even grasping at the surface to fully unravel and understand what is going on.
Well, yes, I think there's loads we don't understand. I think we are moving fast. The big step is to get to a cell. So I don't know, and this is totally off the wall, but I'd say that as I say, I can imagine from a cell developing and optimizing and tuning. Like, you could almost imagine a scenario where if you were to put a slime mold down somewhere in an environment that it's got all of the machinery and all of these interdependencies between its different parts, its DNA and its motors, and it can start to build and start to specialize as it gets there. But you've got to have all of that machinery already there to be able to, in my opinion. How you get from nothing to that is where the challenge is. But on that note, I'd say that we're going to Mars at the minute. We're on the moon. Like, you could easily imagine in the next 100 years, we could land on a planet, sprinkle it with mold or something, and then wait 10 million years. And if we went back, I think there would be all sorts of different stuff. Would that make us a god to that world? I don't know. It might be that someone's been here before us and just put this really versatile cell down, maybe, or maybe the cell has come from sulfurous puddles which are being struck by lightning. But the bit there is really hard to imagine how you get the complexity to make it all work as a machine. And that's the bit which is the hardest bit to grasp. But once you get to the cell, I think it is explainable.
Well, I have.
It's not magical, and it's still like this incredible. There must be a layer of a higher order to this than what we can imagine, in terms of it's not just a single amino acid here or there, and like there's got to be some form of actual coding in the DNA, which is how these are all ran, which might be highly modifiable. You might be able to even imagine compiling it almost on it in the physical presence of its environment or something like that. But there's definitely, it's not a linear trajectory from a sulfurous puddle to us, in my opinion. There are gaps in that, but I don't know what the gaps are and what would fill them.
Yeah, what I see as a red line going through all biology is that codified information directs the operation of mechanical machines, which are proteins. There is always that relation and that state of affairs where information directs not only the making, but also the operation of the cellular machinery. And of course, the question is, how did that state of affairs start? I mean, molecules is one thing, but molecules using and storing information, that is a completely different level of state of affairs. And the question is, how did that come to be? How did it start? I mean, we have DNA, which then is transcribed and translated into making proteins. And these proteins then make the basic building blocks to make DNA and RNA and amino acids and so forth. So that is basically a vicious circle where you have no starting point. You have to have everything together in order for everything to work.
Yeah, I'm not disagreeing with that. That's the bit which is the missing piece in any discussion, because how you get to that complexity and how you get to that, because as much as anything, you've got to be able to replicate that and then improve on it. But so to get into a cell, yeah, I've got nothing, no strong. It's easy. It's actually easier for me to imagine someone else like us, like what we could do to Mars. We could grow stuff on Mars, which is completely new species which don't exist yet, based on just by putting a cell down. I think the cells have got the capability for incredible versatility in their shape and form, which can generate because of this ability to optimize against the physicality and the conditions of the environment. But how you get to it, yeah, that's why I think probably why we're talking.
Maybe we should investigate tardigrades, which have survived in space and which have enormous ability to adapt to different environments, and they can be in marine environments, they can be in space, and they can remove basically all the liquids inside of them, and maybe that would be an organism which would survive on Mars. I don't know, but
so maybe when I'm saying take the slime mold to other planets, you just take some of the tardigrades. Then it's an arbitrary argument. I'm just wondering, like, there's a resilience and a robustness to how all of the machinery in us works, because yeah, it just is. And I imagine that it comes about because you have motor proteins which can generate movement and things with polymerize. So you can generate a machine where it's constantly whirring and doing stuff. And all of that I can imagine in terms of what we know and how it works and how they assemble into structures. It was just how you build that in the first place. So yeah, maybe someone like us or someone greater came 50 million years ago and all the planets just seeded them with stuff. I mean, when I was...
if you had two competing hypotheses, Ben, one would be where intelligence is an ingredient and brings forward all this complexity, and the other is just random unguided natural events. What do you think would be a better explanation of that kind of complexity that we observe in nature?
Well, I think there's a big piece missing, and it's a massive driver of selection somehow. Like there's something like this interdependencies which form. If there was a way to work out, like one of the things we're trying to do at the minute is trying to build simplified systems. We're using these motors, like and these polymerizers to see could it like have interdependency. Like because if you've got a motor at either end and it's pushing and pulling, you can synchronize across the whole of a vesicle or a whole of a thing. So, and then if it could start to, and how quickly that will scale in terms of the complexity. But it's just that there's a higher order to all of this in terms of the complexity. It's the interdependency, which is the big challenge to imagine. But if it turns out that scales really fast, then perhaps. But yeah, so the easiest one for me to imagine is that something seeded with, I don't know, I'm going to go off on a tangent here and sound like a madman, but in terms of, yeah, like that there was a cell and then it evolved in the environment. You put a cell down, it's got this unique magical quality where it can differentiate and adjust and tune to that environment. And we could easily start doing that in the next 100 years. We could go around and put cells. So it doesn't move it moves the goal further away as to what planted seeds down here, but yeah.
I say always that even the most basic or simple cell has inbuilt mechanisms to adapt to the environment, and if it wouldn't have that right from the get-go, it would immediately die. And that, of course,
that's the mistake you're making, because yes, it will immediately die, and that will probably be what happened to 99.9999% of all the variations and permutations, but one which can get a foothold, and then if it's got this ability to very rapidly optimize against that, then you could imagine that you could build complexity very fast, as long as you've got a way to capture that in the first place. Because we've got survivor bias, we only see the things which have worked, we don't see anything which hasn't gone to plan. So if there was a way to have a more selection pressure or some way like earlier on, then maybe there are routes to it. The hard bit is how you build the building blocks in the first place, but maybe that will, if we would find that there was a way that could happen, then maybe all of a sudden it would put it back in that it could just spontaneously originate, and maybe there's a driving force which we can't, we don't know yet.
So Ben, do you believe that evolution was actually a driving factor prior to DNA replication and life starting? Do you think that chemical evolution is actually a proposal which has some reason to be brought into the picture? Or would you say that prior to DNA replication, the only mechanism would be unguided random events, chemicals bunching together and somehow then starting to interact and become more complex to in the end having molecular machines and cells?
So yeah, that yeah, well, as I say, I think that from a cell it's easy. How you get to a cell, I don't know. Perhaps it came from somewhere else where there's different pressures or different things. Like, so yeah, I would say it's almost like a. I think evolution from a cell, but not to a cell. It's not really a satisfactory answer for what you're trying to get. I know you're trying to draw on what this has to be created, but as I say, I could easily imagine that we could create life on other planets in a few years' time. So what's to say that it wasn't just another organism from 10 million years ago or whatever it was, 100 million years ago, who happened to come through before us, and that there is some spontaneous formation of stuff, or in a different environment, there's different conditions, or yeah. Not very satisfactory answer, but it's because at the minute I don't know it. Honestly, if I'm not going to give you a flat answer, I think it's definitely evolution from nothing, because there's big gaps in that. Creation, I could imagine that we could seed life, and I could imagine if there's different environment, then yeah. So what's your view?
Yeah, my view is that God created life. I'm a creationist.
God's a bit of a cover-all term. Like, what does that mean in that context? You mean like?
I'm sorry, I didn't understand your question. Can you ask again, please?
Like God's a way of just. Do you mean like just a creator, or?
Yes, I believe that basically we have two possible explanations, competing hypotheses to explain the origin of the physical universe and life. One would be that intelligence was involved, and one where intelligence was not involved. Yes. And I think this is a key distinction between the entire evolution creationism or atheism, theism debate. And one of the distinct attributes of a creator is that he is intelligent. And I, as a machine designer, I know that by my experience, I can get from my boss the job to make a complex machine for a specific purpose. I sit down, put a drawing on the table on the drawing board with all the instructional information to make that machine or a factory or whatever, and then that instruction goes to the factory, and factory workers, they can read the information, then instantiate that device which I intend to build. Now, this is entirely from the beginning to the end a process where intelligence is involved. That starts from the one that wants to make the device, to the designer, and then to those which read the design and then instantiate the thing, the device. And these factory workers, they know also where to take the materials, to purify them, then to machine each single one, and then construct the machine, bring them together, and then have an interlocking complex machine which has a function. And I use that as an analogy precisely to what we see in cells, where the information stored in DNA is the instructional assembly information, and then that information is transcribed and translated to make molecular machines, which are proteins. And in the end, a chemical factory, which makes all the ingredients, proteins, basic building blocks to get life. And that I use it as an analogy where at first point there has to be an injection of information which is stored in DNA to then having the thing go. And the big question is intelligence a more case adequate and capable explanation of that first genomic information, or is it better explained by a random assembly of nucleotides which then became somehow information bearing, and then the machinery to transcribe and translate it and having then these proteins? And I think if we look inside of a cell, then we see much more and higher complexity and sophistication than whatever we as humans can construct and build. And that is, in my understanding, evidence of intelligent design.
Sure. Yeah. Yeah. At the minute, I'm totally not locked onto any route. I just think there's something big missing, which we don't know. I think maybe we're just, we could be just part of someone's experiment. I mean, it could just be that the cells are just, I don't know. Yeah, sure. I don't know.
I have figured out today the smallest free living bacteria is Pelagibacter rubique, which is one of the most widespread organisms on Earth. It makes up 25% of the marine biological environment. And that bacteria has about 1.3 million nucleotides. And in contrast to Mycoplasma, which has just about 500,000 nucleotides, it can actually synthesize all the 20 amino acids which are used in life. If you go lower, like Mycoplasma, it doesn't synthesize the amino acids, so it has to get them from the host. So I don't think that Mycoplasma is the best model to try to understand the origin of life. I think Pelagibacter rubique would be a better model organism. And then you have to ask yourself, well, 1.3 million nucleotides, how do you get that starting from no genome? And I mean, it's a huge gap, and you are correct. I don't think that anyone has a plausible scientific explanation which is based on naturalistic means. And if you calculate the odds, then it is far beyond what the entire history of the universe would be capable of bringing forward.
Yeah, but that's assuming that there's no selective pressure. Those sort of statements don't really mean a lot, because if you have got something which is suddenly spooling out like big long strings of DNA and there's a heavy selection process, because you can do like random evolution of sequences in the lab just by letting it be messy and just spitting out loads and loads and loads, but with a selective pressure on a good one. So it's not the entire length of the universe. I think those sort of arguments don't add much weight, but in terms of how you get to that, yeah, in terms of how you get something which is going to spool out loads and loads of sequences.
Yeah, you know, there was a stream of Professor Dave, and he was responding to Dr. James Tour, and he said, well, polymerization of nucleotides, hey, that's not a big deal. We have polymerization machines. And I laughed out loud when he said that, because that's just nonsense, because there was no polymerization machine on prebiotic Earth. Of course, you cannot resort to that kind of thing. And of course, in a lab, who makes the experiments are scientists, which are intelligent. So they can actually create the entire process and environment to have these attempts, but these are all things which were not extant prebiotically, right?
Yes. Yeah, I'm not arguing with you. I'm not really disagreeing with you. Right? I think it's a massive open question, but in terms of there's not enough time in the universe, that doesn't carry that weight for me, because if you can get something which can, like, you might imagine an early series of protein which was actually starting to generate big nucleotide sequences, but without any specificity. But then over time they get selected for, like, some persist or some do something. It might have been stability at that time. It might have been. I don't think that's as good an argument as the interdependency of the motors, because I mean, if you have got something where you can start to string together nucleotides, or where amino acids start to get strung together, then they would be self-selecting, because some would be better than others. So that doesn't strengthen any view for me. But the interdependency of all of these different parts of a cell working as a system and being able to divide with high fidelity, now that is a much harder prospect to imagine than how you get the individual pieces. Because even if you could, like, you could imagine you could get the pieces, but how they assemble to have like, there's got to be a selection on that.
Yes, during my investigation, I came to the conclusion that the issue of irreducible complexity, which you just tackled, is much bigger than usually acknowledged. Because, for example, there are many proteins which use iron sulfur centers as reactors, and the cell requires to import iron and sulfur, and then you need all that import machinery, and iron has to be it has to be brought into the right form in order to be imported into the cell. And then these centers, they have to be manufactured as well in a production line process, and then inserted into the protein, and so forth and so forth. And for example, just the import proteins of iron, I studied that. If you have just a small sequence which is different than the one which actually is able to sense the iron, then you cannot attach it and import it into the cell. And if you don't have iron...
Now. At the early stages, it might not have had that. Now, the reason that fails is because the incredible specificity which has been optimized around that. So back in the early days, like, it may not necessarily have had such stringency, and it might not have had such a high... like, there's an optimization which has gone on. So I don't know if the iron's uptake and the mutations and stuff now is a good readout of what would have happened in a simpler system. But again, I'm not arguing that what you're saying is wrong, just that all of these proofs aren't as good as the interdependency proof. I think that's the hardest one to overcome for me. How you get all
I mean, let's suppose DNA replication. the smallest bacteria, they use about 30 proteins, and one is helicase. Helicase alone is a protein with about 1,000 amino acids, and if you don't have helicase, you know what happens. You cannot untangle the DNA strands and then replicate the information. So how do you get helicase prebiotically? I think these are big open problems and questions.
Yes. So yes, no, I mean, yeah, totally. There's a lot we don't know, and there's a lot of assumptions, and a lot of, it's not always clear what people believe and what they actually know in a lot of these discussions, and there's a lot of gaps in whatever argument you're going to try and make for this. Yeah. But one of the things
I think like, basically you can have three. Yeah.
So changing the subject slightly. So
yeah, I think you can have. You can have basically three positions here. You can say, well, I believe chance did it, or chemical evolution. Someone like me can say, I think intelligent design is the best explanation. Or we can withhold judgment and say, well, we don't know, and maybe in the future science has some explanations.
No, and as I say, I can evolution from a single cell. To get to that, I don't know, but I'd imagine like space biology, or maybe something coming from somewhere where it's a totally different environment where this sort of stuff, or it might have been created somewhere, like we could just be nothing more than a petri dish on some PhD student equivalent's desk in but one of the things which is interesting in terms of, like, I guess with because obviously the creation evolution thing, but in terms of like humanity's role in all of it, like, because if you're religious, then I guess you have the mindset that humans are special or unique, or how do you sit on that? Because one of the things which is very clear from this is that all of the cells have got memory molecules, these Talin molecules. They've all got the same, like, down to quite far down, they've all got synapses, they've all got like neural activity of some sort going quite far back. Like, how do you see that in terms of where are we exceptional, or do you see it as where do you sit on that?
Well, the question is, of course, what is the type of information that Talin stores, conveys, transmits, signals? And that is precisely what John is asking here. I don't know if you are already that far to investigate this. Have they been able to identify the syntax of the Talin codes? So basically, what kind of information is it that it conveys? What does the code do actually?
So the model which we're trying to develop is that so what's known in a single cell, like a cancer cell in a dish, is that these Talin switches, they coordinate the behavior of the cell, what it's doing, and instruct it how to migrate or to stay put, and how to signal and to reprogram and all of that. And in the brain, you've got trillions of neurons all making these synapses where they're coming together. Sorry, let me find and they're talking to each other. So in that these cells are all just talking to each other and making like connections. So you have an input into one cell, which then signals to another and updates it, and then that one then propagates that through the network. So in terms of neural nets and all of that type of stuff, it could just be that these switches are being updated, and you've just got this, like, almost like what we have optimized with our computation, where you just adding up this information to go around these circuits to connect all of the inputs of the organism to all of the outputs of the organism via a computational module, which is the brain and the nervous system. But in all of those scenarios, even into a slime mold, they're experiencing something, processing it, and then doing an output. So at which point on that gradient from a mold or beyond through to us do you class as being, do you have the exceptionalism of a lot of religious people on humans being this unique thing, or do you see it more scientifically?
Yeah, I have really no answer to the transition point from consciousness, intelligence, sense perception to what is purely physical, like neurons responding to a signal from the senses, and then the sense perception which is perceived through my consciousness. Like, for example, if I eat a piece of chocolate, then I have the sense perception of the smell of the chocolate in my mouth, which then is transmitted to the brain, and there that information is somehow processed, and then I have that perception. Now, what is purely physical and what is based on consciousness, or if it is everything just purely physical, I am not sure, but I think science doesn't have an explanation in regards of that question, which is basically the problem of consciousness.
No, I'd agree. And like, if maybe all of the inputs, like if you taste and you smell when you eat a bit of chocolate, it's feeding into a circuit. Like, and I always think about like, yeah, a mathematical representation is what I tried to call it in the MeshCODE paper, where all of the inputs and all of the outputs are connected via this complex circuitry of ones and outs. But that same complex circuitry is in fruit flies, it's in worms, it's right the way down, just not as complex. It's not got as much parallel processing, as much circuits, and much but to me they would have a similar level, like a simplified level, but they would be, I would predict that they'd probably be conscious. I don't think there's a point where something suddenly we become exceptional. And I know that a lot of people I've spoken to in the past who have got more of a religious tilt of this sort of idea that humans are somehow exceptional in that. And I just wonder what your view on that was.
Yeah, so John is asking here, you had mentioned signaling to the nucleus. Do you think it has relevance to alternative splicing commands?
Yes, absolutely. We're working on that at the minute. These signals which come in, growth factor signals coupled with mechanical signals, and we can clearly see that the alteration in the splicing and the altered variation in the genes. So yes, and the same with the transcription factors and the other regulators. They're moved around the cell. So if they're in the nucleus, you'll get one set of splice products. If they're at the adhesion, obviously they're not going to be altering the splicing of the mRNA, so you're not going to get that. So yes, they definitely are connected with the splicing and the transcription, and with the epigenetics and which genes are pinned open and which ones are shut down type thing.
Well, that is very interesting, and I can imagine while you were speaking that eventually this is one of the systems which are involved in adaptation, where you get a specific signal from the environment which is received on the surface of the cells, and then signaled inside to the cell, processed, and then depending on what the signal is, the cell will express a certain kind of proteins which will be a better adaptation to that kind of environment, like temperatures or whatever the difference is.
Sure, yeah, I agree. And I think also there's a lot we don't know about, like it's changing really fast, but in terms of the genes and then the rest of the DNA, maybe there's actually information of a much higher order than what we currently understand in those regions. And maybe that's like where all of the actual optimization occurs, and the genes are just the objects which the machinery of the cell's using and calling and modifying. So I think we're scratching, I think we're moving incredibly fast to understanding this, but we're still, I think in 50 years time we'll look back at what we thought now, or certainly what we thought 10 years ago, and just chuckle to ourselves.
Would you agree that there is a distinct difference between information which is processed by cells and organisms, which comes from the outside of the environment, than information which comes through nutrition? If you eat something, then the organism will digest and have a different response to that food which is ingested. And based on that, it will react, it will adapt, it will do what biologically our bodies do. But then there is a complete different kind of information, which is we as humans actually convey, which is like, for example, we are talking now each other, me to you, you to me, and tonight I will remember what we talked about. But yeah, but that is kind of information. I don't think that that kind of information is somehow, somewhere stored inside of cells with epigenetic codes or whatever. What do you think in regards of that?
Well, I think it absolutely is, and I wouldn't even draw the distinction between what you eat and the physical environment. How I picture all of this is that this is all just cell signaling. Like all of my cells are talking to each other to make these complex organisms, and when I talk to you, I'm sending signals to you, which is triggering in your ear into your nervous system. But then the shapes of these molecules, these talins, changing the shape, changing the activity of those individual synapses to send information to different circuits of the brain. And just so I think everything is, which we've discussed today, is written into the shape of the talin molecules in the brain, which then overnight lead to slightly more longer lasting changes, which might be the epigenetic changes or the bigger wiring thing. But it's all cell signaling, and that's why a synapse is so beautiful, because it's two cells making a really intimate contact where they can just knock a signal through to the next one and update that cell. So that's where I think that maybe like cell signaling might just have way more complexity to it than what we currently imagine, and it might stack really high, because if it's exponential, the number of cells which are talking to each other, then very quickly you get to levels of information content and permutations, which is way beyond what we could ever imagine. So I think it's
there have been some theories like that information is stored in microtubules, things like that.
Yeah, so I would say on that, the good thing about the MeshCODE theory and what we're trying to convey is that these molecules changing shape, we can physically see it, and it's quantized into either an O state or a 1 state, and you've got these strings of information. The issue with the microtubules and the quantum, entering the quantum world, maybe that it's true, maybe there's something going on there we can't understand, but it's really hard to imagine, and it requires, that's like way beyond, like how you can have wet quantum effects is really hard to imagine for me. Whereas here, these are physical interactions. These are like well mapped out. We can recapitulate it in a lab with a single molecule. We can recapitulate it in a lab with a cell, and we can build it up, and we can show that this happens. And we can show it happens because we can use mutants or drugs or things to manipulate this in a cell and show that it happens.
Right.
So,
right.
And I think that's the big trick, because all of the stuff we need to do, you need to, like, we're trying to understand how life functions. So we always take it right back down to the single molecules and then try and build up the complexity back. And then human mutations, we can see how they affect the behavior, the property of the molecules, all of that. And it's moving across the levels of explanation from genetic to the molecules to the machines, the cells, etc., which is the key, I think.
Okay, there is another question. Have you read anything of the papers on the multiplexed code theory application to biological codes, specifically the genetic code? If so, do you think the same concepts would apply to Talin Code?
I haven't read much on that, so I don't know much about the multiplex code theory, but the idea we'd have was that these molecules changing shape is really, really quick, and you can encode it. You can switch a motor on and flick a state. So you can do that instantaneously, as quick as it takes to pull on it. And then that will hold for a specific length of time. But then that will integrate with the genetic code because of this mechanotransduction and because of this talking from these molecules to the nucleus to give rise to longer lived changes and basically reprogramming of the cell as a function of the signals it's received. So in that respect, it'd be multiplex between a relatively dynamic encoding of information coming in, causing longer term changes, and in the brain, strengthening of some synapses to others, sending the information differently between those different cells. So I guess it's multiplexing across levels, but I don't know if that's the same as what John's talking about.
Now, as I said previously, I have collected about at least 45 different epigenetic codes, and I don't know of any besides maybe the Glycan code, maybe, we know that the structure of the glycans attached to the protein on the surface of the membrane, they store information. Then eventually also the histone code, the sequence of amino acids on the histone tail. But besides that, I don't know basically of any other known ways how the codes and information is stored through all these other epigenetic codes. Now, you said that the different states of the Talin code is just limited in time, and I don't think that it can actually freeze, let's say, its state of affairs and then be like some kind of memory. Is that correct? So it changes, it signals, and then it goes back to the previous state of affairs. And then that means that the information would be just conveyed for a short period of time. Is that correct?
Well, I think there's two ways. I think it can be very fast. If you have a molecule on its own, it can fold and refold. If you hold it still, it will, in a permanent environment, it would stay like that forever. But obviously it's not perfect in the brain. I can't which way is up or down on this. It's really weird. It's mirrored. But there's loads of things which decorate onto this, which lock it in specific states. So I think the adhesions in our arms or in our organs aren't particularly dynamic. They lock down with all of these proteins. So on top of the talin, you've got 250 other proteins interacting, fixing those patterns and determining the output. So yeah, this would be like a RAM, but also a computational thing, because you would have this dynamics. You could update this and then change the whole cell's behavior. And I don't know if I can share something else. Just give me 1 second. The computer's really not. The computer knows it's tea time. So this is a poster which we've got coming out soon in Journal of Cell Science, but this is what I wanted to just show, is that this is a computational device, these adhesive structures, because you can see that, because if you take a stem cell which is like able to differentiate into lots of different lineages, then you can trigger it to differentiate into different cell types just by changing the stiffness of the ECM. So if you make this jelly, it turns into a fat cell or a brain cell. If you make this glass, it turns into a bone cell. So these are computational devices, because they grab onto that matrix, probe it, pull on it, and see what, and then that sends signals into the nucleus, because this is total reprogramming of that cell. It's switching on and off different paths to cause the cell to fully differentiate with no other factors varying. So just by changing the mechanics, which is why we know that these are computational devices, these adhesions, and the talin is the main this main linkage between them. So where that cell's grabbing on, it's this network which assembles this computational device. So we start to imagine that these adhesion complexes, where that cell's physically grabbing on, they have a certain pattern of information in which is let's go back to that slide. So basically you've got 13 switches, and depending on the system, whether it's moving or whether it's still, then you've got them in a certain pattern, and then you can move them and change the pattern by either moving outside the cell or switching the motors on, or both. But you constantly update that information in them to lead to those global changes by whether you have the transcription factors or the splicing factor here at the bottom of the cell, or able to go into the nucleus. So it's computational, definitely computational, and that's mapped out. That's not a speculation. That's experimentally shown.
And these other molecules which you mentioned, you said I think 150 or so.
Yes.
Are they also? Is it possible that amongst them there are others which are also information or storing or transmitting or information bearing or information storing? Could it be that there are different systems which interact in an interface together somehow?
Yeah, absolutely. I mean, the vinculin. So as this goes more into the one state, you recruit more cytoskeleton, more linkages, more connections. The signaling changes. These get bigger. They have a different signaling output, and they recruit different doses of those molecules, because you can start to imagine that depending on the exact number of talins and the exact number of each switch, you move these molecules around the cell. So what we're starting to get to is this notion that, and this might be where we make a bit of an advance on this, is if you look at a cell in culture on a plate, then it never becomes ordered, because it's never in contact with all of its neighbors and with the perfect surface. So it all looks really chaotic and really noisy. But once the cell is in contact with its neighbors and the correct matrix and getting the right signals, I think that this has very specific, highly selected patterns, which then instruct the signal to the nucleus and switch on the gene. So I think there's a level of order to these systems which we don't see in the current experiments, or we glimpse, because it's not we don't see them. It's just it's masked by this loss of the higher order system.
Well, I think this is a massive amount of information through a variegated system of different epigenetic information systems which work in a joint venture together to instantiate all the complexity that we see. And yeah, yes. I mean, the open question, of course, the philosophical question is, how did all that complexity come to be? Some prefer to say it's evolution. We as creationists think that there had to be a super intelligence involved to instantiate all this kind of complexity. Yeah. Now, Ben, unfortunately, I will have to go. I have to
end. No, that's good. Maybe you could finish just with the question of where do you see, like, in terms of humans in the general evolutionary development of organisms, like, do you see humans as exceptional, or do you see us as part of a gradient, a spectrum of complexity?
Well, I think we distinguish ourselves principally through the fact that we can use intelligence, very complex communication systems. We use words. We can instantiate things which are far, far more sophisticated than any animal can instantiate in the animal world. We have free will, which animals don't. Animals, in great general sense, they operate based on instincts, while we are not. So, of course, there is a complete, entirely different level of complexity involved here. And even if we look in regards of the human body, I mean, each single cell of us stores 2.3 billion proteins, which is absolutely unimaginable. The multicellular organism, which is composed of 37 trillion cells, and they all signal the cells even on long distance together. And I mean, this is something which we are not even grasping the surface of.
They absolutely do, the gorillas do as well, or the monkeys, or like the dogs, or the mice, or they all have this. The only difference is the computational power, like the circuitry has got, like, you could easily imagine that there'd be a modification which would make, we could get, like, it wouldn't take much if you've got an exponential improvement in memory and in processing power. Like if you look back at your old computers or your Game Boy or whatever, I mean, it's just because of the sheer computational power, which is just because of the complexity of the emerging circuits. So I see it as on a gradient, and easily we could be superseded.
And we know that now that neurons, they are far, far more complex than initially thought. These are really computers which process tens of thousands of signals at the same time. They are microprocessors in a literal sense. So the more that we actually dive and investigate and unravel what happens in biology, the more we see new levels of complexity which were not expected
exactly
before.
But again, you skirt over this year because the C. elegans worm, which is 1 millimeter long, has got nerves, it's got synaptic connections, and it's got the same machinery, just with like whatever it is, 300 nerves instead of a trillion or whatever. But it's the same machinery, it's just with the lack of the computational power. But in terms of from a religious perspective, like to me, there doesn't seem to be a huge distinction apart from in the complexity. But I get the impression that that's not how you see it.
Yeah, I think that your work is fantastic. I think that you are unraveling.
you should politics. You dodge the question really well.
Yeah, I mean, to really understand the distinction, as I tackled previously, there are things which we distinguish ourselves from primates, which is the ability to have abstract thought. And being able to compute and do things that animals can't. We have free will. We have a sense of morality. We know what is right, what is wrong, and so forth. So I see actually a big gap. I don't see a gradative transition from primates to humans. I as a creationist, I believe that God created us upon his image, knowing what is right, what is wrong, being able to communicate on an entirely different level that animals are able to. So I think all this is actually evidence that there was a creator involved to create us and the animals. Okay, Ben, I much appreciate for you coming to my channel, to my stream. It was a very pleasant conversation. I hope that we can eventually keep on having a dialogue, or to email if I have a question or so, if maybe someone of the audience has a question. And I'll keep looking forward for what you unravel in your forthcoming studies and investigations.
Thank you, and it's been fun.
Okay, thank you very much. Thanks for the audience. See you next time. Bye-bye.